The following content is provided under a Creative Commons license. Your support will help MIT OpenCourseware continue to offer high-quality educational resources for free. To make a donation or view additional materials from hundreds of MIT courses, visit MIT OpenCourseware at ocw.mit.edu.
Okay, good morning, everybody. Morning, good morning, good morning. All right. We're going to continue our discussion about cancer today, as well as on Friday. In between, you have an exam on Wednesday. Partly in preparation for that, I'm having office hours tomorrow, between 3 and 4 in 76453. And as the note says, come with questions that you have about immunology, which I taught you, and the introductory cancer class that I taught you. If you have questions about other subjects that will be covered in this exam that Professor Siv covered for you, it's best to go to her office hours or to your TAs or the other sections that will be held. All right, so towards the end of last lecture, we talked about smoking and cancer, and I warned you against the evils of smoking, and I hope you were paying attention. This slide shows you some pretty startling statistics that relate the increase in smoking, shown here, among men in this country, which began around 1900, and you can see rapidly increased over the first part of the last century. And you can also see that about 20 years later, lung cancer rates rose equally quickly. And this was some of the evidence that smoking caused lung cancer. And we now know, as I mentioned to you last time, that there are lots of carcinogens in lung cancer, in cigarette smoke that cause lung cancer. Lots of mutagens, either mutagens in their native form or pro-mutagens that can be converted into mutagens that we expose ourselves to through the process of tobacco smoking. That was men, but lung cancer has also increased in women. You can see this also precipitous increase in lung cancer among women in this country, and lung cancer has now passed breast cancer as the leading cause of cancer deaths among women in the United States. You can see that in the case of men, smoking levels actually started to drop quite some time ago, and coincident with that, lung cancer deaths among men in this country also have been dropping. It's a direct result of smoking cessation and people not starting to smoke. That had not happened in the case of women until very recently. In fact, just last month, a study was published that showed that also for women, lung cancer rates are now starting to go down, and that's a direct consequence of the fact that fewer women are smoking. It takes a few years to see that effect play out because people who were smoking 20 years ago are getting cancer based on that now. So examples of why smoking is bad for you. Cigarette smoke can be considered an environmental carcinogen, although it's one that we expose ourselves to. I talked to you about a variety of other environmental carcinogens that we expose ourselves, that we get exposed to. I also described this phenomenon by which chemicals that are not in their native state mutagenic can become mutagenic through metabolism in our bodies. Pro-mutagens can be converted to mutagens in our body. And I showed you the example of benzoapyrene, which is in cigarette smoke and gets converted into a mutagenic form in our bodies. And there are many other such. There are also examples, and I didn't say this explicitly, of non-mutagenic carcinogens. This goes against the rule that carcinogens are mutagens. These are actually non-mutagenic, but they're cancer-causing, so we call them carcinogens. And examples of this would be alcohol. And asbestos. These affect cancer rates, we think, because they cause tissue damage. Alcohol in the liver, for example, causes tissue damage in the liver. Asbestos, if you breathe it in, can cause tissue damage in the linings of the lungs. This tissue damage then results in increased proliferation. Cells are recruited to grow in order to repair the damage that was caused. And this can then indirectly result in mutations. Because as I mentioned to you, every time your cell divides, there's a chance that a problem will happen, a chance that a replication error will occur. There's a chance that an important gene relevant to cancer will incur a mutation. So more replication, more proliferation, the greater the chance that a problematic mutation will take place. I didn't say this explicitly, but likewise, problems in chromosome segregation can occur every time a cell divides. We have intricate systems to ensure that chromosomes separate properly so that each daughter cell gets the right complement of chromosomes, but sometimes that breaks down. And sometimes cells get the wrong number of chromosomes. We talked about this in the context of meiosis. But non-disjunction events can occur in mitosis as well, leading to chromosome imbalances, which are also hallmarks of cancer. As I showed you last time, if you look at the chromosome content of a cancer cell, it is often very, very abnormal. And these abnormal chromosome numbers occur due to defects in chromosome segregation, including non-disjunction events. Okay, so various things that we expose ourselves to or that just go along with the normal process of cell division can lead to mutations. And as I emphasized in last lecture, cancer is the consequence of the accumulation of mutations in critical genes. So to summarize what I told you about last time, we now think of the process of cancer development as going from a normal cell through multiple steps to the development of malignant cells. This process, for most types of cancer, will take years to accomplish. Cells acquire these alterations over multiple years. And these arrows represent mutations. Mutations to cellular genes. Mutations in processes that are important in determining the normal behavior of these cells and allowing these cells to behave abnormally. This process unfolds over time, as I mentioned, and gives rise to this phenomenon, or this hypothesis really, for which we have great evidence now, the so-called clonal evolution of cancer, which I drew on the board for you last time. This is a nice version of that same concept from a figure from a book from Bob Weinberg, who teaches this same class in the fall. Here you have a row of normal cells. Within these cells, a mutation arises. This mutation gives that cell the ability to expand, perhaps better than its neighbor cells, so that a number of daughter cells carrying this mutation are born. And within this now clone of singly mutant cells, a second mutation can arise, say in this cell here. This mutation likewise confers upon these cells a greater ability to grow, divide, survive, outcompete their neighbors. So you get a lot of these cells too. And then within that expanded clone of cells, a third mutation arises, and so on, until one has enough mutations to have a fully malignant cell. So what are the genes that are relevant here? Well, let's think about the processes that we know are important in cancer development, and those are listed in the bottom right. Proliferation is the most obvious one. Cells proliferate abnormally in cancer, so mutations in genes that regulate proliferation are likely to be important here. Cancer is a disease of cell number, too many cells. You can get that because you have too much proliferation, but you can also get it if you have too little cell death. So mutations in genes that regulate cell death processes are also found in cancer. I told you last time about angiogenesis, the process by which blood vessels are recruited into the tumor. These too are recruited by virtue of signals that the tumor sends, some of which are the product of mutations. I told you that cancer cells move in the process of metastasis. They break their interactions with their neighbors, and they begin to move throughout the tissue and ultimately throughout the body, so they have increased cell motility. And they also can invade. They can invade through the basement membrane. They can invade into the local tissue. So they have increased invasiveness. And there are other changes that take place within cancer cells. And so these mutations then collectively increase proliferation. Decrease cell death. Increase angiogenesis. Increase motility. And increase invasiveness. It's important to know about these because today we're able to target some of these mutations. We're able to target the gene products formed by these mutant genes and thereby create better therapies. So our goal is to understand these processes such that we can ultimately control them more effectively in the context of treatment. Okay, well that then leads us to what are the genes? What amongst the 22,000 genes in your genome get mutated in the development of cancer? How can we find them? Nowadays, we sequence the genomes of cancer cells. That's how we find them. But that's not always been true. It's actually been true for only the last few years. And so I'll give you some examples of how they were found previously. And why do we care? Well, I've already indicated some of this a few minutes ago. But the reason we care to understand the disease at the molecular level ultimately we'll be able to provide in very accurate detail improved diagnostic information, improved prognostic information. We'll be able to tell that an individual has cancer by detecting abnormal genes in their blood, circulating DNA in their blood, carrying specific mutations associated with particular cancers. We'll be able to diagnose the disease at an earlier stage that way. We'll be able to figure out exactly what mutations are present in the cancer cell to know whether that's a tumor that's going to go on to kill the patient or sit there and do nothing. Should we treat the patient aggressively or should we leave them alone? Only with this molecular information will we be able to figure that out in detail. We will, and we already, use this information to make better cancer drugs, molecularly targeted therapies that will replace conventional chemotherapy, which I'll teach you about on Friday. Chemotherapy can work, but it's highly, highly toxic. And we'd like to be able to get rid of it and replace it with drugs that are much more specific, much more selective, and much less harmful, except for the cancer cells. And this will usher in a new era which I would argue is already here in some small way, called personalized medicine. The individual's disease will be diagnosed at the molecular level, and a specific therapy will be designed for them based on those alterations. We'll dial up the right therapy based on that information, and again, hopefully, yield better results and less toxic side effects. Cancer is leading the way, actually, in personalized medicine, but it'll be true for lots of diseases in the not too distant future. Okay, so what are the genes? Well, I'm going to introduce you to two broad classes of genes today, the first of which are called oncogenes. Onco for mass genes, oncogenes, cancer genes, cancer-causing genes of one sort or another. The first of these oncogenes was identified in the context of oncogenic viruses. For example, Rous sarcoma virus, which is a retrovirus. We'll learn more about those next week. Retroviruses are viruses with an RNA genome. HIV falls into the same class. Rous sarcoma virus, or RSV, has been studied since about 1910 or so. It was discovered by a virologist at Rockefeller University by the name of Peyton Rous. Peyton Rous was a virologist, and a Long Island chicken farmer came to Peyton Rous with a prize hen from his collection, a prize hen that had a tumor mass growing on its breast muscle. And the farmer brought this hen to Peyton Rous and asked him to cure the hen because it was very valuable to him. Peyton Rous took the hen. The farmer said thank you. The farmer went away. Peyton Rous then promptly killed the hen and isolated the tumor, and was able to isolate from the tumor a virus that could infect another bird and cause cancer in that bird. So this was the first example of a virus associated with tumor development. There have since been many examples of viruses associated with tumor development in animal species, a few examples in people, but relatively few. Most cancers in humans are not virus associated, but some are, like human papillomavirus-associated cervical cancer as an example. But these viruses studied in laboratory animals were extremely important in teaching us about how cancers arise normally. It was known using Rous sarcoma virus that if you took a normal chicken cell, fibroblasts from a chicken, and infected it with Rous sarcoma virus, it would cause those cells to round up and begin to proliferate abnormally. And these cells were given a term called being transformed. Transformed cells that had the appearance of cancer cells. After that work, a great deal of effort went into figuring out what were the genes of Rous sarcoma virus that allowed the virus to cause those cells to become transformed. And it was determined that the virus carries a single gene called SARC, S-R-C for sarcoma, which is responsible for this transformation process. You could basically just add the SARC gene and the same process would occur. Trying to understand the origins of the SARC gene, two investigators at UCSF, Mike Bishop and Harold Varmus, in around 1975, determined that the SARC gene has a homologue, a related copy, in chicken cells. And they went on to hypothesize correctly that Rous sarcoma virus stole this gene from the chicken cells that it was infecting and incorporated it into its genome. So the SARC gene, this cancer gene, had a cellular origin. Moreover, they were able to show that SARC exists in human cells. And this was quite shocking. This cancer-associated gene was present in our DNA. This discovery led Bishop and Varmus to win the Nobel Prize in 1989. And a funny story happened that day. This is a true story. I actually worked for Varmus as a PhD student, so I heard it from the horse's mouth. Varmus's sister-in-law was standing in a cafeteria line at Berkeley waiting to get her lunch, and two guys were standing in front of her. And one guy said to the other, "What are you going to have today?" And the guy said, "I don't really know." And the first guy said, "Well, don't get the chicken, because two guys just won the Nobel Prize for showing that chicken causes cancer." This is sometimes how the public perceives what we do. But anyway, they went on to win the Nobel Prize for this important work. And they proposed that there were genes in our DNA, which they referred to as proto-oncogenes, which could undergo mutation and become oncogenes. In the case of RSV, the mutation was to take the gene out of the genome and stick it in the genome of a virus. But as we'll see today, there are other ways to do that too. Now, what the heck are we doing with proto-oncogenes in our genomes? What are these genes doing there? Why do we have the SARC gene? Why do we have other such genes? Who can tell me? Why is it beneficial to have a cancer-causing gene in our DNA? What might these genes be doing? Yeah, normal metabolic processes or perhaps normal proliferation. Our cells divide too. You go from a single cell when you're at fertilization to 10 to the 13th cells. It's a lot of cell division. That's a controlled process. There's lots of genes that are devoted to teaching your cells what to do when they're supposed to do it. And these genes are presumably involved in those normal cell division processes, but they get corrupted in the context of cancer. They get altered so they don't work properly. Okay? So RSV was the first example. SARC was the first example. But it still led to some skepticism, some concern that, in fact, what was seen in the context of these viruses might not be relevant to real human cancer, which, as I told you a few minutes ago, rarely involves viral infection. And so along came Bob Weinberg. Who was and is still at MIT. His lab is in the Whitehead Institute. And Weinberg did a critical experiment. He started with an individual. Sorry about that. This is a person, sort of. And this individual had bladder cancer. Weinberg isolated the DNA from the bladder cancer. To ask the question, were there cancer genes in there? Were there oncogenes in there? Were there mutationally altered genes which were the reason that this tumor arose? And so he did an experiment. He took this isolated DNA and he introduced it into some cells in the laboratory. These were immortalized mouse cells. Immortalized meaning that they would grow in the lab forever, which, by the way, is not normal. Normally, you take cells out of your body or out of a mouse, put them in the lab, they'll grow for a while, but they'll stop growing eventually. These cells were immortal. They could continue to grow. But they were otherwise pretty well behaved. They laid flat on the dish. They had normal boundaries between cells. They were non-transformed. They didn't look like cancer cells. and they were non-tumorigenic. If you introduce those cells, these cells, into an experimental animal, they wouldn't cause a tumor. Okay? They were immortalized, but they were otherwise pretty well behaved. He introduced the isolated DNA from the tumor cell through a process we call transfection. Where basically the DNA gets sheared up and introduced into the cells, such that, roughly speaking, each cell is getting individual genes, spread out amongst this population of mouse cells. And then he waited. And what he found was that at low frequency, whereas most of the cells stayed in their normal morphology, occasionally he got this. Can somebody get that? A transformed colony of cells. And he assumed, correctly, that this colony arose because one of these genes was a cancer gene that allowed these cells to divide abnormally. He further could show that if he took those cells and introduced them into an animal, they would now cause a tumor. So they were not just transformed, they were also tumorigenic. He went on to isolate the human gene. Which was not difficult to do because the cells themselves were mouse. And so he was looking for the human DNA sequence. And he found eventually that it was a mutant version of a gene called RAS, a gene that you've actually learned about already in class, and I'll tell you more about in a second. This discovery made by Weinberg's lab and a few other labs at the same time in the early 1980s is the reason I'm standing before you. When I was your age, Weinberg came to my college and gave a lecture on this work. And I was so excited about the potential of learning about cancer at the molecular level that I decided right then and there to start working on cancer, and have been doing ever since. So sometimes the things you learn in class actually change your life. Not to say that's going to happen today, but sometimes it does. Okay, so Weinberg isolates the RAS oncogene from these bladder cancer cells. They went on to sequence the gene to determine how it was different from the normal copy of RAS, and that's illustrated here. Here's the normal RAS sequence. This is now not working. Here's the normal RAS sequence. It's a version of RAS called HRAS that's insignificant. You can see the codons, the encoded amino acids. Here's the change that has taken place within the cancer cells. They didn't have a G in this position. Instead, they had a T in this position. This gene wouldn't encode glycine like it's supposed to, but instead valine at codon position number 12. This change, this single nucleotide change, this single amino acid substitution, caused this signaling protein to go from a regulatable state, as shown here, and hopefully this is familiar to you because you learned about it already in the signaling part of cell biology. RAS is a GTP binding protein, which normally cycles between an active GTP-bound state and an inactive GDP-bound state. It goes from on to off through this hydrolysis of GTP. In the context of this mutation, the GTP hydrolysis is inhibited. So the protein stays in its GTP-bound active state. It gets stuck on. And rather than signaling in a regulated fashion, signals constitutively. Rather than telling the cells to divide when they should, it tells the cells to divide always. And that's presumably why this mutation is selected for in this type of cancer and many, many others. Okay, I remind you that RAS is in a signal transduction pathway. Here's RAS itself. RAS interacts with upstream receptor molecules and growth factors There are intermediary adapter proteins that help that interaction. There are downstream signaling proteins like kinases. And ultimately, there are nuclear transcription factors and target genes that they regulate. This is a signaling cascade. And actually, what we learn in cancer is that many of these genes, not just RAS, but many of these genes can be mutated in the development of one or more cancers. For example, some genes like these are amplified, and I'll tell you more about that in a second. Others are the product of translocation, so that they're expressed abnormally. Others have structural mutations like deletions, and others have very subtle mutations. The RAS mutation is a subtle mutation. It's a single nucleotide change that allows this gene to function abnormally. Okay, so importantly, oncogenes dominantly transform cells. Weinberg added a single mutant oncogene, and it transformed those mouse cells. The mutations are gain-of-function mutations. They're dominant mutations, gain-of-function mutations. There are now about 300 or so, and the number is growing, known oncogenes. Within the 22,000 genes in your genomes, about 300 of them can be converted to an oncogenic form through mutation. What types of mutations do we find? Well, I've listed them on that slide, but I'll just write it down as well. Subtle mutations. RAS is the classic example. A single amino acid change will convert the protein to an oncogenic form. We can also find gene amplifications. Your genes are present at 2 per cell. One from mom, one from dad. You're supposed to have 2. Sometimes in cancer, amplification of regions of the DNA occur. So you go from having not 2, but 4, 8, 50, 100 copies of the gene. And you can imagine having too many copies leading to too much protein product would actually lead to inappropriate signaling. So here, the structural gene may not be mutated. The amino acid sequence may be the same. You've just got too much of it. Also, genes can be rearranged. Gene rearrangements. For example, translocations. And I showed you some pictures of translocations, chromosomes that get joined inappropriately together. This can break 2 genes and form a new gene in the context of the translocation. Perhaps a gene is expressed from a weakly acting promoter, normally. But because of a translocation event, a very strongly acting promoter gets stuck in front of that gene by mistake. And now the gene is expressed at very high levels, inappropriately. It's not unlike the consequences of gene amplification. So translocations likewise occur frequently. And we'll hear about consequences of amplification and rearrangement next time when we talk about therapies. Okay, so oncogenes act dominantly. They can transform cells all by themselves. But this should be in your minds creating confusion. Because I've also told you that cancer is a multi-step process. And if cancer were a multi-step process, how could it be that single mutations can transform cells? But in fact, we now know that single mutations are typically, and maybe never, are not sufficient to produce true cancer. So given that, can somebody explain how the Weinberg experiment worked? How was it that Weinberg was able to transfer a single gene into these cells and cause them to become transformed and tumorigenic if single mutations aren't enough. Why did this work? Anybody? The key is that they weren't normal cells. The cells he started with are already abnormal. They were already immortalized. They'd already been growing in the laboratory dish for a long time. So they were sensitive to a single mutation, but normal cells in your body are not. And that's a good thing, because as you sit there today, you probably have about 1 million RAS mutant cells in your body, scattered around. And that's just based on the normal mutation frequency. The likelihood is that we all have about 1 million or more RAS mutant cells in our body. But because RAS mutations are not sufficient to drive cancer formation, that may be in a tumor-initiated cell, but it's not yet a full cancer. Other mutations have to take place thereafter. We now believe that there's probably something like 3 to 20 mutations are required to make a full-blown cancer. Okay. So I've told you about oncogenes, and I've related their function to normal cell division. And that's appropriate to do. Normal cells do get signals to divide, make more of themselves, and moreover, they get signals to stop dividing when it's time to stop. In embryogenesis, once you form the liver, you want to stop cell division within the liver cells. When you wound yourself, you recruit cells to divide, but once the wound is healed, you want it to stop dividing. And so there's complementary signals, stop signals, that come into play to cause the cells to stop. Cancer cells have defects in both of these classes of signals. We've been talking now about the oncogene signals. They are the go signals, the signals to tell the cell to divide. And because of these alterations, like in RAS, the cells are more capable of dividing. They make more of themselves. Moreover, the brakes on this process, the stop signals, are also typically lost in the development of cancer, such that now the cells lacking the braking signals will continue to divide still more. And these two classes of genes are called oncogenes, that we've been discussing already, and tumor suppressor genes that we'll discuss from now to the end of the lecture. Tumor suppressor genes. There are a number of tumor suppressor genes now as well. More than 200 known, so that's 500 or so known cancer genes already. The first one, the very first one, described occurred in this type of tumor. This is a child with a tumor of the eye. The tumor is called retinoblastoma. And based on the examination of the genes within those tumor cells, it was hypothesized that the retinoblastoma gene that was responsible for this disease was one of these tumor suppressor genes, before any of them were actually known. That led ultimately to the cloning of the RB gene, the retinoblastoma susceptibility gene called RB, also in Bob Weinberg's lab, which encodes a protein called PRB. And further work in Weinberg's lab and many others showed why cells get rid of the RB gene in the development of that cancer as well as other cancers. The RB gene is now, or the protein that it encodes, is now known to be an important regulator of the cell cycle. We learned about the cell cycle. Cells go from mitosis to G1, into S phase, and then G2, and then into mitosis again. This is a regulated process, and the RB gene, or protein, is important in controlling it. The RB protein acts here by blocking the transition from G1 into S. And I'll tell you a bit more how it does so in a moment. This is when the protein is in its active state. It can be inactivated through phosphorylation. A phosphate group, actually many phosphate groups, can be transferred onto the RB gene through kinases, which are stimulated by growth-promoting signals. Growth factor binding to a cell will ultimately result in the stimulation of these kinases to phosphorylate the RB protein, taking it from this active state to its phosphorylated state, which is inactive. So the brake is released because the kinases inactivate the brake. Okay? Just a little bit more detail about RB. A lot is known about this process now. This transition from G1 into S requires some transcription factors called E2F transcription factors. And RB binds and inactivates the RB transcription factors, keeping them silent when it's in its active state. When it's in its active state, RB has a little pocket to which the E2F transcription factors bind. Here's PRB, and here are the E2F transcription factors, and they are sequestered by the RB protein and can't otherwise do their job. But when RB gets phosphorylated by those kinases, these phosphate groups interfere with the binding of E2F, and E2F is then liberated to carry out its normal function, driving this transition. Okay? So this is how the brake functions, and it's a very, very important brake. RB is mutated in a very high percentage of cancer cells, not just retinoblastomas, but many others as well. And the pathway that it is controlled by is likewise mutated in a high frequency. Tumor suppressor genes, like RB, are the brakes. They negatively regulate proliferation. As such, are these genes normally hyperactivated or inactivated in cancer? Hyperactivated or inactivated? Inactivated. That's in contrast to the oncogenes, which get activated, hyperactivated. These get inactivated or lost. These inactivating mutations are recessive. They are loss of function. Again, in contrast to the situation with oncogenes, which are dominant mutations, gain of function. And this actually creates an interesting challenge for us. If these are recessive mutations, and a cell acquires a mutation in one copy of the RB gene, and I'll just show the chromosomes on which RB exists, happens to be chromosome 13, we have a normal cell. Say it's a normal cell in the developing retina, and that cell incurs a mutation. Random chance, cigarette smoking, probably not in this child, but random chance leads to the inactivation of one copy of the RB gene. What's the phenotype of this cell? It's normal. These are recessive mutations. Having one normal copy is sufficient to provide RB protein, to provide control of the cell cycle. So the cell is normal, but the cell is predisposed. Predisposed because now it only has one functional copy left. We could call this the first mutation. And now, within this clone of cells, if a second mutation occurs, then we're in trouble. And this second mutation can occur in one of a couple of different ways. For example, it could be that by random chance, bad luck, the normal copy on the other chromosome gets mutated. Or it could be, and it's actually more frequent, that a chromosomal event takes place. For example, a cell arises which only has one chromosome 13 through chromosomal non-disjunction, and it's the one with the mutant copy. These are functionally equivalent. Both of these cells are RB deficient. And they are on their way to becoming cancers. Tumor suppressor genes therefore require 2 hits. Two hits, 2 mutational events to inactivate the 2 normal copies of the gene. They can be 2 mutations within the gene, or they can be 1 mutation within the gene plus some chromosomal event. And I've shown you one example here. There are others. Okay. So that looks good. Everybody gets it, right? No problems? Let me show you this picture, which I actually showed you on the first day of class. This is a child who has bilateral, multifocal retinoblastoma. This child has 12 different retinoblastoma tumors affecting both eyes. Not only that, this child comes from a family in which retinoblastoma is passing through the generations. The grandfather had it. He passed on the predisposition to multiple of his children, who passed on the predisposition to multiple of their children. This is an example of a familial cancer syndrome. Familial predisposition to cancer. Most cancers don't have this kind of pedigree. Most cancers are sporadic, but about 10% of cancers look like this, with a clear family history. Familial retinoblastoma, familial breast cancer, familial colon cancer, and there are others. These are caused by mutations in genes like this. This individual that I showed you, the individuals in this family, have inherited a defective copy of the RB gene from one of their parents. And as such, they have this genotype. They are heterozygous for an RB mutation. Heterozygous for an RB mutation. And because they're heterozygous for an RB mutation, they are highly predisposed to developing retinoblastoma. And the reason is that within their cells, within the developing retinal cells in their body, 2 mutational events are not required. Instead, in them, all of their cells look like this. All of their cells are in that heterozygous predisposed state. And therefore, in them, a single hit is required to give rise to a cell that is lacking the function of RB altogether. And that's why they're predisposed. And that's why BRCA1 patients get breast cancer and why APC patients get familial colon cancer. This slide also raises for you some interesting questions, and we'll talk about them next time. Here's a person who has the right genotype, but he didn't get the disease. Why not? And another question, what would happen if two individuals who were heterozygous for a mutation were to marry, have a child who was homozygous for an RB gene mutation? What would happen then? We'll talk about that next time.